to today's webinar, Taking Control of Food Spoilage and Safety in the Era of Food Microbiomics. My name is Karen Whitman, and I will be the moderator today. Before we get started, I have just a couple of housekeeping slides to go over with you. This webinar will be recorded and will be available in 1 to 2 days. We will also have a question and answer session at the end of the webinar. So if you have any questions during the webinar, just go ahead and put those in the queue. Go ahead and change the slide. Okay, and then here are some steps on how to put questions in the queue. So in order to do that, you'll see down at the bottom of your webinar handle, there's a section that says questions. Go there and then click on questions, and then you can put your question in the little text box and press enter, and it'll go right into the queue so Greg can answer it at the end of the presentation. It is now my pleasure to introduce to you Dr. Gregory Saragusa. Dr. Saragusa is Senior Principal Scientist with Eurofins Microbiology Laboratories at Eurofins. His former positions include Senior Principal Scientist with DuPont Danisco, Director of Poultry and Food Safety Research for AgTech products with similar success at organizations such as the USDA Agricultural Research Service, as well as the Universities of Nebraska and Georgia. In addition, he has served as an editorial board member for the Journal of Food Protection and Applied and Environmental Microbiology, while also serving as an ad hoc reviewer for several other peer-reviewed scientific journals. His research and expertise has spanned the fields of intestinal, veterinary, and food microbiology, detection methods, and probiology, among many others. Dr. Saragusa obtained his Bachelor's of Science and Master's of Science degrees in microbiology from Louisiana State University and a doctorate from the University of Arkansas. He has over 100 peer-reviewed articles and book chapters and has presented numerous abstracts and talks. He has served in leadership roles with the International Association for Food Protection and the American Society of Microbiology. Aside from research in the U.S., he has extended activities to Russia, Denmark, and Kenya. His latest endeavors focus on the application of genomics to food microbial ecology. It is my pleasure to hand over this webinar to Dr. Saragusa.
Thank you, Karen, and greetings, everyone. Very happy to be here, and I look forward to some good discussions at the end of this little talk. So I'd like to welcome everyone to the new era of food microbiology. Food microbiomics is exploding in the microbiology field, and there are reasons for that, and I'll go through some of those. Also, today I'd like to get you more familiar with the terms that are used, because we hear a lot of genomic terms, and the definitions of those aren't always very clear to people who don't work directly in those fields. To start with, though, we're going to use the word microbiome quite a lot, so I'll give a definition. The microbiome is the population or community of commensal, symbiotic, starter cultures, or pathogenic microorganisms that literally share a space. That means a space of a sample, that means in an environment, that means in water, and as you'll see, there are many other microbiomes that are being developed and discovered as we speak. So I'd like to ask two questions, two different slides. Can you relate to this? If your job is to perform a plate count on a sample, or if your job is to order that analysis to be done or to direct it, how do you do it? The normal steps are you homogenize things, you plate them, count and record the results. And if you're in the laboratory, you might pick up that plate and look at it and ask yourself, you know, these colonies sure are pretty or interesting looking. I wonder what they are. And then normally you toss that slate in the autoclave bag. You get rid of it. What if your job is to find out what is causing some problem, fill in the blank, whether it's gas formation, slime formation, high plate count, whatever the problem is, and you use a cultural approach. Go through the same process, homogenizing, plating, counting, and recording, and ask yourself, which of these organisms on this plate might have caused that gassy defect? Or why didn't an antimicrobial that we added work? Was one of these organisms resistant? Well, asking those questions through the lens of a plate count have given us a lot of information. Plate counts are great indicators. But what if you take it a step further and your job was to identify colonies on one of those plates? Let's just take an example of how that would proceed. If there are 100 colonies on this plate, and you picked and isolated all of those colonies and sent it in for identification, at about $75 per identification, that's $7,500. Okay, but that's from a single auger, and that's from a single growth condition. What if you're also interested in the bacteria that come from anaerobic growth, or psychotrophic growth, or lactic acid bacteria media growth, or anaerobacteria, etc.? You can see real quickly that to get a picture or a profile of organisms in a sample and identify them with a name is quite difficult to do by cultural microbiology. You probably all know this. So the key question I'll ask 2 times during this talk is, how well do you know your product's microbial populations? And the answer might be that, well, we know them pretty good because we've done functional plate counts. We've grown things on lactic acid bacteria media, or on standard methods agar, or on potato dextrose agar, and we have counts. All right. But the real question is, do you know the names of the makeup of that population? Do you know the citizens of that population? Well, plate counts are a wonderful tool. Cultural microbiology will continue to serve us, but there's something about plate count that we should all understand and probably have heard in our background or education, and that is that plate counts will only culture a very small minority of organisms. In fact, this is called the Great Plate Count Anomaly. This term has been around for a long time, and if you just glance at the figure on the left, Figure 1, you see it's mostly gray, and that indicates levels of bacteria, the darker it is, from a direct microscopic count. On the right is the plate count of that same sample by culture. You see there's a huge difference. Also, even if organisms grow on media, here's an example where 3 different media were used, and if you look at the very bottom, the number of genera, genus, or species isolated is about the same. However, if you look up in the middle of that table, you'll see, even though the number of genera was the same, they were different depending on the media. So it doesn't really do you much good if you're only using one type of media. So what do? Do you use every type of media under every condition to get a profile and identify all of those bacteria individually? I think you can see real quickly that you're going to run into a problem of feasibility. Well, now we have tools that allow us to amplify, sort, count, and compare genes on a large scale in a short turnaround time, relatively inexpensively. And what that tells us is who is there, what microbes are there, and what groups do they belong to, and in what proportions. That's the new era of microbiomics. There's been an explosion in research I mentioned earlier. This result was actually just taken yesterday, 22nd of June. Querying PubMed online, there are almost 22,300 publications that have a keyword of microbiome. And if you look at the bottom half of the slide, we see a trend. And for those of you who use Google Trends, you understand what this means. You see the blue line. Microbiome biology is ever increasing. Why? If this is so great, why haven't we done it? Why haven't we been doing it? A couple of reasons. One is the cost of sequencing has finally dropped to a point where it's doable. Secondly, the number of genes that are deposited in databases has increased geometrically. Those two things have allowed us to use the technique of microbiomics. Microbiomics is widely applicable, I said earlier, to many different sample types. And this gives you an idea of some of those different ones, from soils to insects to food animals to food, as we'll see, to forest soils, grasslands, human GI tract, human samples, etc. You can look at the list. So virtually any biome or any niche on Earth has its own microbiome. And that microbiome can now be explored with a tool that we did not have just a few years ago. Microbiomics even has its own journal. Several journals, in fact. This is just one example. So I get back to the key question. Using the lens of the plate count, how well do you know your product's microbial population? So I'd like to go over a few terms before we move on. We've already talked about the microbiome, that population or community of microorganisms. The microbiome is determined by sequencing marker genes of those bacteria and fungi, for example, that are on that sample. Microbiomes can include identity in the relative proportion. It's not an absolute quantitation, nor is it a substitute for pathogen detections. Marker genes are genes that serve as name markers to identify an organism. They are highly conserved. That is, they don't change much over evolutionary time. The most common marker for bacteria is the 16S gene. You'll see that written many different ways. And I should emphasize that these terms are going to be used with slight variations, and it can be a little bit confusing. For instance, the 16S gene can also be written as the 16 small s, or the 16S rRNA, or the 16S rRNA DNA. The point is, the 16S gene is a segment of DNA. It codes for ribosomal RNA, that's its product. But the 16S segment of DNA also serves to give names to bacteria. Okay, 16S rRNA sequencing is a process of amplifying those genes in a sample. Another term you'll see is metagenome. That's the study of microbial communities in complex samples by sequencing all of the DNA, not just 16S. We'll see what that means in a few minutes. That's also known as shotgun sequencing. NGS stands for next generation sequencing, also known as high throughput sequencing. Taxonomy, when they talk about that, if you remember from Biology 101, we use a system of classification, and I have an illustration there. You can remember the names of the taxonomic levels by the mnemonic device. King Peter came over from Germany seeking fortune, standing for kingdom, biome, class, order, family, genus, species. And fortune is strain type. Okay. Then you'll also see the acronym Operational Taxonomic Unit. That's a classification of microbes. It's almost synonymous with species. All right, so not to get bogged down in terminology, I just wanted you to be aware that some of these terms you'll see in different forms. Don't be alarmed by that. I think you'll get the idea. Hopefully by the end of this talk, you'll have a good grasp of what some of these mean. So the 16S marker, there's a model of it there on the right, is found in bacteria and archaea. Archaea, if you remember, used to be classified with bacteria, and they're actually their own different taxon, and it's one of the three major branches of life. Yeast and molds don't have the 16S marker. They have something else. We use a marker called the IPS marker, or the 18S marker for yeast and molds. So talk about how you did plate counts. How do you do plate counts? We go from the sample to a growth step, counting colonies to calculating. Identity, I put a box around that because you might or might not do it. It might be that all the information you think you want is on that plate. For instance, how many organisms will grow at a lower temperature? That's the cyclotrophic count. How many lactic acid bacteria can we estimate that? The LAB count, etc. And how do you do biomes? I have a couple of slides that will illustrate this. The general steps are pretty much the same no matter who's doing it. Sample is homogenized. The matrix can be separated by filtration. There's a lysis step where DNA is isolated, the total DNA from that sample. It's put through a semi-purification. You'll see in a minute that we can amplify the 16S gene. We go through sequencing of all of those genes, and then we do the bioinformatics. Bioinformatics is the collective term for all of the analysis that goes into studying sequences. Bioinformatics is its own field. There are several journals of bioinformatics. Colleges and universities worldwide are offering degrees in bioinformatics. It's its own field of data analysis. Some of the programs that people use that you might hear, programs like Chime, Mother, other proprietary softwares. There are a lot of softwares for doing bioinformatics, but it doesn't have to be mysterious because many of these analytical tools are actually open source and online. So, to determine a microbiome, there are two major methods right now. The 16S targeted method, which is what we'll focus on, or a shotgun metagenomic method. Very briefly, the 16S targeted method starts with food. DNA is extracted. The 16S gene is amplified, and you can see that there's a change in the ratio of the tan-colored DNA to the background, the vast background of blue DNA, which is food. We change that ratio by amplifying that 16S bacterial gene marker. Then, in the sequencing laboratory, those DNAs are barcoded, which is part of the process. And then it's sequenced. It goes to bioinformaticists for analysis, and the result is a profile. This is a pie chart. There are many different ways you'll see of displaying this graphic. Pie charts are probably one of the most commonly used. Every wedge on the pie is a different proportion of a group of bacteria. Very briefly, the shotgun approach is different. The shotgun or metagenomic approach is different in that DNA is extracted. There's not a targeting step. We don't just target the 16S gene. Instead, everything is sequenced. And there's some advantages and disadvantages to using targeted versus a shotgun approach. But the end result is, if you're looking for bacterial profiles, you'll get a result that gives the relative proportions of bacteria. But I wanted you to understand that there are two ways of determining the microbiome of samples. So how do you go from a gene to a name? So we've already been through that. You lyse cells, you extract DNAs, you amplify that 16S gene, you have a sequence, and then that sequence gets sent to the bioinformaticist who will take it and query databases. That is, they'll take that sequence that you see there in the upper left and put that whole sequence into a database and ask it, which organisms by name have that corresponding 16S sequence? What can you do with that information? First of all, it gives you a name. You can identify those citizens of the population. The database maintained by the U.S. National Center for Biotechnology Information is called GenBank. It has millions of deposited sequences with the corresponding names of organisms. You can determine the relative proportions, and you can determine ecological things like the intrasample variation, the intersample variation, the total diversity. Probably one of the most useful things, though, for you as food scientists is you can compare spoiled pairs. That is, you can compare two samples, one of which is spoiled, one of which is not. I would also add that in the process of doing the microbiome, about 75% of that procedure, time-wise, is tied up in the bioinformatic analysis. That's changing as we talk. Currently, it's a very involved process, analyzing the sequence. So I want to talk a little bit about how we can incorporate microbiomes into state-of-the-art current food microbiology. Right now, if you have a sample, for instance, I've listed a few here on the left, and you culture those, I put in parentheses, this is a semi-targeted approach. Semi-targeted because depending on the agar you use, that will be what the culture method tells you. If you use PDA agar, you'll probably get yeast and mold. If you use enterobacterial agar, you'll probably get enterobacteriaceae, etc. It's semi-targeted. It's not specific for any one organism, but it's specific for groups of organisms. In the microbiome approach, it's targeted. You can use that information. You can use those names to do a couple of things. One is you can identify specific organisms that might be problematic or that might tell you something about your process, that indicates something. And you can take that and make a specific assay for it. So a specific assay could be in the form of a PCR assay, or it can be a more refined or targeted culture method. So instead of looking for every group of organisms, every organism that will grow on an enterobacterial plate, you might look for specific ones. So now you're focusing your cultural indicator microbiology. Who would use this information? Just about anyone in food production, in the food production field, can have a use for this information. If you're involved in food quality and you want information about spoilage, shelf life, if you want information to write your HACCP plans, safety information, preservation efficacy, all of these can be determined from a microbiome. If you develop new products and you change the ingredients, that's going to affect the microbial populations that eventually grow in that product. If you're responsible for obtaining raw materials from different geographies have different microbiomes. And if you want to know what those are, you can use this approach. And you really can't do that with a cultural approach. And maybe most importantly, and I'll discuss a little bit more toward the end of our talk, is that the FDA, the USDA, and EFSA are all investigating the use of microbiomes as part of the regulatory framework we work under. So regulatory science is doing research on this right now, and it's being done in a very, I would say, a very systematic and scientific way. So these regulatory agencies are investing in understanding how we can use microbiomes to help producers make food that is more stable and more safe. So what can you do with all that? Well, I listed 10 uses here. I'll just go through them very quickly. If you're asking questions about spoilage, why is your shelf life short? What if you want to know what microbiome is associated with samples that are consistently pathogen positive or pathogen negative? What if you have a particular fermented food flavor profile and you want to catch that and preserve it and find out what it is, what microbial profile is actually giving you that flavor profile? What if you have area and plate counts that are consistently out of range? What if you seem to have organisms that persist in your environment and are recalcitrant to sanitation? What if you want to know desired organisms in a starter culture, if you're a fermentation producer of starter cultures? What if you want to find out sources of contamination, for instance, from raw ingredients? You can use that information, as I said, to sharpen your culture, to sharpen that indicator sword. And then what about predicting, for instance, salmonella and fresh-cut chicken meat, or some other product, or gassy spoilage or off flavors? You can use this to determine what communities of bacteria are associated or not with that defect. So it enhances our power. Will this do away with cultural microbiology? No, but it greatly empowers the tool. Clay County is still a staple of food microbiology testing, and a lot of it is done every year. That isn't really going to change. So at this point, we're about halfway through, and I'll just ask, keep track of your thoughts and questions. If you have pencil and paper at hand, jot them down so that you don't forget them when we get to the end. So here's some examples. I'm going to run through some text-stated examples as well as some examples from the literature. These are 3 examples from the field, and I'm just going to state them. One was a producer that had a high APC of deli salad. Had this problem for many years, in fact. They changed sanitation, they changed and bought new equipment, never solved the problem. Turns out that through the microbiome approach, backed up with culture, that it was found out to be an ingredient that would randomly bring in organisms that contributed to this high count. We determined what those organisms were, what the ingredient was. The company then went to that raw ingredient provider. Another case, a processor was having issues with sporadic gassy spoilage of salad dressing. Performed the microbiome process, found out that actually there was a sanitation flaw in one of the lines that contributed a fungal component or a yeast component to that contamination. In another case, working with the researchers who were developing ways of sanitizing food plants, they were very interested to know what is the microbial profile of floor drains that seem to be correlated with Listeria presence or absence. All of these things could not have been determined feasibly by culture alone. So here's in this next series of slides, like this, are examples from the literature. So here's one example of a traditional product. This is actually a product made in Korea, and it's a fermented, semi-dry soybean cake. And we can see that we have, in the pie chart, we have a large component of Bacillus sonorensis. Here's the pie chart. These are, on the bar charts, are different samples of that product. And we can see that it's composed of several different major bacterial groups, not something we can determine by culture. These are microbiomes of American Coolship ale, more or less similar to Belgian ales, if you're a beer aficionado. And what the workers did here was to show not only the diversity of bacteria, but also the diversity of bacteria by different batches and over time. This is a study comparing the microbiomes of vegetables. And if you pay attention to the wording here, you'll see that on this lower line, all of the words are ending in EAE. And that indicates family. You can go with the same information up to the order. So using that same information, we can go up the taxonomic tree from family to order to class, or down to genus and species. And then you can compare those profiles and ask questions like, how does this profile in spinach compare to a profile in bean sprouts? Not something you can do like culture. What about surfaces? Here's an example of public restrooms, and what we see is that they group in different, more or less in some different groups on a PCA plot. But what's important is that these names, for instance, this red organism here, Propionibacterium, is not a surprise. It's very commonly associated with skin. What about in kitchens? And think about your food plant when you look at these kitchen data. Well, we can see that the areas that have the highest number of OTUs, which are taxonomic groups, bacterial species, if you will, those are areas that have the most traffic, and that makes perfect sense. In front of sinks, in front of a stove, in front of a refrigerator, makes perfect sense. But we see that there's diversity in those areas where we have more contact, and we can identify what that diversity is. Again, not something you can do by culture. The FDA has a science group that's studying microbiomes of different samples and environments and foods. And these next two slides are from that group. They studied the bacterial groups, and here we see families, EAE, and how those families differ on one tomato plant, from the roots all the way up to the top, depending upon whether that leaf was exposed to sunlight or if it's the underside of a leaf. You have a different microbiome. So the microbiomes here differ greatly. Just look at the diversity here in colors. Again, not something we can determine by culture. But the question was, do any of these specific pie communities correlate with a higher or lower incidence of salmonella? I want to give one example with fungi. These are the same samples, and instead of targeting the 16S gene, they targeted the ITS gene and published on the diversity of yeast in fungi. Very difficult thing to do by culture. Through microbiomics, they were able to not only do it for one sample, but do it for multiple samples. This is a comparison of milk cheeses, like Croatian ewe milk, sheep milk. And again, here's a bar chart. We can see diversity over time in between cheese types. And then you can use that same information to create a Venn diagram and ask which of these organisms are common to all three cheeses, which are common to two cheeses, and which are individually associated with one particular cheese. Applying it to pork sausage, this was a really interesting study from the University of Nebraska and an industrial partner, where on these samples they determined the microbiome, which was largely composed of lactic acid bacteria. Here they're identified down to the species, but also they did plate counts, pH tests, and sensory characteristics. So the goal here was you can associate a particular profile with the development of off flavors over time. If you did this by culture, all you can do is glean information from these 6 different media, but you would not know the identity of what composed that APC count, that psychotropic count, and our bacteria coliform and E. coli. This is a very recent study, and I put it in here because many people drink coffee, and many people have these coffee pod machines, and this just goes to show you what you can do with this tool. This is the little tray underneath the machine that catches the liquid, and it was done on different days. And just look at that diversity from left to right over 60 days. It illustrates one point, that not all sequences that are determined have a name. Here it's listed as other. This is going to lead to new discoveries of new organisms that just have not been cultured, so they have not been studied up till now. Again, here's another one of the coffee pod machines, and it gives you an idea of the diversity if you take that sample from different parts of the machine. A lot of others there. So we revisit these questions. What are those colonies? What is their identity? Which groups of organisms are causing my problem? Or which organism is causing this problem? Or which organisms were resistant to an antimicrobial? Now we have a tool. And combining that with a cultural approach is a very powerful tool. Eurofins started doing next-generation sequencing and microbiome analysis about almost a year ago. Eurofins has been a leader in genomics for over 14 years. We are a complete service organization when it comes to genomics. That is, we have most of the major technologies for sequencing, Illumina, PacBio, and different instruments, state-of-the-art machines for doing high-throughput sequencing. We also have state-of-the-art extraction chemists, bioinformaticists, and finally, people who can use that data with you to solve problems. Eurofins Microbiomics right now relies mostly on the 16S microbiome approach. All of this information is available online on the web. You can certainly ask more questions about it, but it'll give some examples, and you can use this information to share with your colleagues and to study and recall some of the things that we talked about today. I would want to bring out one point here, is that amplifying the 16S gene can be done in different ways. The 16S gene is about 1,500 base pairs. And so you can amplify different regions of that 1,500 base pairs. And so within our service offering, we can offer you different sites to amplify. And it'll give you more or less information about the organism. So it isn't just sequencing the 16S gene in general. Using this approach, we can look at specific parts of that gene and learn different things. So I'd like to summarize that microbiomes now let us see that uncultured part of our environment, and in this case, our foods and our processing environments. This has often been called the uncultured majority because, as I said, the estimate is that only about 1% of organisms on Earth have been cultured or are culturable. It's only when we know the citizens of our food products and environments that drive shelf life, quality, and safety can we design assays and culture methods or methods to mitigate their presence. So microbiomics combined with culture is a good guide. Our FDA, FSIS, and CDC are moving toward a genomics-based genotyping approach as we speak. This will be linked to microbiomics. Eurofins offers not only full microbiomics services, but partners with you to use our food science expertise and our scientists and the collective knowledge of our group to help you utilize that knowledge in solving the problem. So a couple of takeaways. The United States and other countries are investing very heavily into microbiome initiatives. Food is certainly one of the most important microbiomes, in my opinion, that we can determine. But they're highly varied because our foods are highly varied. So there's a lot of work to be done. Also, if any of you are attending the IAFP meeting this coming July, August, we have a roundtable discussing more of this. So we have several experts from the FDA, from academia, and industry there to talk with you and to try and answer questions that you bring with you and to discuss new ideas. So please, if you're at the IAFP meeting, we would love to see you at the roundtable. So at this point, I will stop and entertain questions that you might have. Thank you.
Thank you. That was a great presentation. I will now go ahead and take questions out of the queue. So if you have questions, be sure to enter them in the queue. Again, that's through the webinar panel. It's at the very bottom. There's a section that says questions. Click on that and enter your question there. Greg, your first question is, how would you envision our regulatory agencies using microbiomics to protect the food supply?
FSMA stresses preventative measures, and both the FDA and the FSIS are looking for what might be considered those microbial profiles that are normal. That is, versus an aberrant state. That state might be high indicators or pathogens. So this is not a futuristic question. It's currently being researched. I would say that our regulatory agencies will utilize microbiome data, in my view, from four main factors. One, that data hopefully will come from inputs from the industry, certainly from academia and other R&D entities, and demonstrating its ability. So that's the first thing. It's inputs from industry showing how it can be used. Secondly, advances in sequencing and bioinformatics technology is going to get the price of sequencing down even further. And the prediction is that it will be so inexpensive to perform that the next step will be to develop new and better at-desk bioinformatics tools. So instead of having to send everything off to a bioinformaticist, perhaps much of the data generated can come to you and you can analyze that with new tools. So the technology is changing. It's going to be different. Thirdly, sharing results to assure FSMA compliance, I think, has to happen. And that way, that cooperation will usher in, hopefully, a new era of indicator microbiology as well. So again, we're not going to get away from culture. And then fourthly, the use and acceptance of microbiomics will be fostered by industry, and this powerful tool will lead to an academic response to train new scientists in its theory and practice. And at that point, you'll see it become more common.
Okay, thank you. The next question is microbiomics accepted by FDA, AOAC, and BAM?
AOAC, and what was the last one?
BAM, BAM.
Okay. To my knowledge, at the moment, our laboratories have ISO 17025 accreditation for conducting genomics. So that includes microbiome analysis. Those agencies, though, to my knowledge, as of yet, don't have it formally accepted. But as I said, there's a lot of research going on, and I would expect to see that certainly within the next couple of years. Whether or not it will be accepted as or used as a tool in the BAM manual, I would predict that that's going to happen. I don't know when.
Okay, thank you. The next question is, does this microbiome technique work equally well for bacteria and fungi, or is it just less accurate for fungi? And then the second part of that question is, does the microbiome technique work equally as well? Nope, never mind. That was just one question. Here, I'll ask it one more time. Sorry about that. Does this microbiome technique work equally well for bacteria and fungi, or is it less accurate for fungi?
The databases that we use to query have a preponderance of bacterial sequences. The database for fungi, they exist and they're growing, but they're smaller. So given the set of sequence data for fungi, being able to assign a name to that sequence, it's possible that you're going to have several sequences that don't have a deposit in the database because the database simply isn't as large. In the case of bacteria, it's ever growing. So we have to get the fungal database, yeast and mold, to that point, to that same point. Okay, is it less accurate? I think the accuracy is going to be in part a function of the database. So in that respect, it's going to be maybe not less accurate, but perhaps less precise or less broad. Maybe that's a better way to put it. Extraction is a very important part of this, and I mentioned earlier that Eurofins has some very much expertise, leading expertise in extraction from foods. Some bacteria are harder to lyse than others. Some fungi are more difficult to lyse than others. So they can be different, and if you have differences between those two, the accuracy is going to be different between bacteria, yeast, and mold. I think as the databases grow for yeast and mold, we're going to see more and more use of it.
Okay, thank you. The next question is, how big of a sample is needed for a typical microbiome test?
Say that again.
How big of a sample is needed for a typical microbiome test?
A good sample to shoot for is 25 grams, not unlike culture. Then that can be used to dilute to some degree, either 1 to 10, 1 to 5, or even greater, 1 to 100. That's certainly enough. The amount that's actually extracted is considerably smaller. So it's very important that the initial sample be representative, because there's going to be a small subsample taken from that for extraction. I would say you can shoot for 25 grams, but it can be as small as a swab, as a cotton swab that's used to, or a sponge that's used to swab surfaces. And sometimes those volumes might only be 1 mil. And we can certainly concentrate those bacterial cells and get DNA out of that.
Okay. The next question is, with something like a probiotic that claims to contain multiple strains of various organisms, could a microbiome testing be useful to ID and ensure all the organisms are declared are actually in the product?
That's a great question. Yes, a microbiome analysis can be used to discern strains that are added, or organisms that are added. But I want to make it clear that microbiome analysis, as it is now, will bring you down to the genus or species level. And I mentioned earlier that below species we have subspecies or subtypes. Most microbiomes, as they're done now, don't necessarily get down to that subtype. So if you have a probiotic that's a mixture of very similar strains, you might not get the resolution you need. There are other molecular biology techniques to do that. If you envision, though, that you use shotgun sequencing and you have longer reads of everything that's there, so longer than 1500, but you have approaching the whole genome, then you should be able to segregate and detect the proportions that come from each of the bacteria that you've added. That's a good question.
Okay, thank you. The next question is, the microbiome determined is from DNA, but doesn't necessarily mean that the bacteria is visible. So again, I'll ask that one more time. The microbiome determined is from DNA, but does that necessarily mean that the bacteria is visible?
Visible or viable? I think maybe the question meant to ask if just because you detect a signal doesn't mean there's a viable cultural cell or a viable cell associated with it. If that isn't your question, please email and we'll address that one. But is all of the DNA from necessarily viable cells? No, it's not. And that's a question that affects all microbiome analysis. But I will say this. Whether or not the DNA comes from viable or non-viable cells depends upon the state of the DNA when it's extracted. If it's been exposed to different processes that destroy that DNA and does things to it that make it non-amplified, then you're going to get a different result. But there are ways of doing a community analysis that we didn't talk about here today that let you look at only DNA, or I'm sorry, only nucleic acid from viable cells, and that is to measure mRNA. So it's not DNA, it's RNA. Eurofins has state-of-the-art RNA nucleic acid chemistry analysis. Measuring the mRNA profile of a sample, while a little bit more involved, isn't as commonly used, does give you a better estimate of the profile that comes only from viable DNA. In order for a wedge to show up on the pie chart, there has to be a pretty significant number of cells in the original sample, probably about 10 to the 3, 10 to the 4. Just depends on the organism. So to have that at one point, at some point in the life of that sample, there had to be cells that were viable. And I think that's pretty accepted amongst the ecology, the microbial ecology field. A very good question.
Okay, so our last question for today is going to be, hold on one second. Have you studied LM in the environment using this method? And if so, have you seen any of the other Listeria species that particularly often associate with LM? Example, often see Inocusa, when you have Listeria monocycrosis on a swab?
First question, to answer that is, I don't have data here to show you about that. That's a question that people are working on, about whether or not a specific profile can be associated with Listeria presence or absence. So I would say to you that that is forthcoming. The second part of that question I didn't quite hear, but I do want to say that it brings us to the point of, it's very important to understand, if you're looking for that association, what is the correlation between a profile and, for instance, Listeria, that you have a concomitant accurate pathogen test that you're doing. And microbiomes can also be used on enrichments. So if you have a cultural enrichment and you're going to plate that and go through a process, or do a PCR test on it and go through a process of determining presence absence, you can also profile that enrichment. And that will tell you some very good information about what it is you're actually enriching when you're trying to look for Listeria that might be there in low levels. Karen, there was a second part to that question. Did you want to repeat that?
Yeah, I can do that. So it just said, have you seen any of the other Listeria species that particularly often associate with LM?
Okay, all right. I think I answered that in the first part. All right.
Yep, I think you did too. So those are our questions for today. Thank you everyone for tuning in for this webinar. We will be sending out a recording of this webinar here in the next 1 to 2 days. So watch your emails. Again, thank you everyone, and thank you Dr. Saragrosa for your expertise today. Have a good day, everyone.
Bye, everyone.